Hello? Morgan, hello.
Hello.
Hey.
Oh.
Jesse was having tech problems.
Oh, okay.
Any better here?
Yeah, there you go.
Are you?
Okay. Hey, Morgan. Yeah, I'm happy to dive into other stuff. I'm setting up the notion. Let's see. It is the 66th meeting of the Cognition Futures Reading Group. Wow, that's something. And it may be. I don't know if we're going to meet again this year or not. We can talk about that. But really, appreciate everything. I'm really looking for. I'm super excited for next year. Not to be like hype train and Mr. Sunshine pumping rainbows and whatever, but like I'm really, I can't wait to just like get all this like current stuff out and like kind of recenter and focus on next year. Like there's been a few talks, like things we talked about Saturday. There's been some movement in the on or else Discord's Big Ideas Discord about whoops, I'm showing the wrong screen apparently. Well, now you know what to do before you arrive on campus. Sorry, let me change that. Here we go. Anyway, I'm very excited about what's ahead. There is I don't know if I can easily show this. I guess I can. Yeah, I'll put this here. I think you can see it here. In the Big Ideas Discord, there's some stuff here. I made a new thread to kind of I made a new channel, and I'm going to start putting. Oh, I haven't finished submitting it yet, but you can see ones that are in the works. For this, I like machine learning for diagnosis, neurodivergence, autism, medical applications. I know Bradley started computational medicine here, so thank you for kind of jumping in on that, Bradley.
All right.
I think that'll be an exciting thing to do, building off of this and other things. There's a lot of things here, like my analogy stuff there, diverse intelligences, cybernetics. Please, what I guess I might suggest is under these project ideas, and I'll make one for cybernetics. We already have sort of a medical computational medicine and things like that. I kind of want to make this channel for like project development specifically, like pushing the projects forward. And I'm doing other admin and organization work about helping structure that basically. I've taken some inspiration looking at I looked at this again, and I feel like I don't think it's changed since I last looked at it, but this is a really nice document. I feel like a lot of people come to the lab and maybe lead or just glance over, but there's a lot of like interesting thought put in here into why things are the way they are, and like kudos to Bradley for doing that. This was sort of the vision paper here. And that's kind of inspirational. Some other things I'm trying to do that will make all of these things make a little more sense in the future. So that's kind of my general update on that. Yes, finals. Yes, fatigue. Yes, excited for next year. Yes, everything else that you'll already know. I think that's my update. So I'll turn it over to if Amanda or Bradley wants to say anything, or Morgan.
All right. Thank you, Jesse. Yeah. So, yeah, Amanda and Morgan, do you have any updates?
Sorry, just fixing the internet here.
Yeah.
So just not sure what the most relevant ones are. Just some nice developments on the organoid front, or at least in terms of getting access and lab space and some of the services that UCSC seem to be providing. UCSD is interesting, obviously, for Alison Motry and the Bradley Wojtek and their early paper on the electrophysiology of organoids. But it's great to see that the UCSB group that has probably the most recent kind of endogenous oscillations of cerebral organoids paper. One, they're a part of the Brain Engineers group, so that's cool. In that this is the kind of UCSC led consortium of it's mostly UC groups, but one at Washington University, and I checked with Logan, and he doesn't know that group, but he was like, but I was planning to get in touch with him about something I'm working on, so that was cool. He now has 2 reasons to get in touch with him. And yeah, you can check. Sorry, my parents just arrived here, so my daughter's excited. Yeah, so UCSC is kind of leading this effort to provide live culture in the cloud, and provide it as a service for classrooms, and hopefully for other labs too. So hopefully it can be something. I assume it's kind of modeled on the Allen Observatory. In the sense that we're doing something here that's kind of hard to do. And so let's try and give people the best kind of remote access to it, right? So if you check the Brain Organoids channel, you'll see, UCSC just had a press release from, Tuesday or something, maybe Monday, and talking it up, and I'm gonna definitely reach out. So Katerina is the grad student that we met with previously, Bradley. Yeah. And I see now that the paper that she's first author on, the last author is described in one of the other, you'll see I took a snippet out of their press release. It's like he's the bioengineering guy that's involved. So he's not listed under the team in the Brain Engineers website, but he's the one who's doing a course spring 2024 that's called Math of the Mind or something like that. So which seems like super cool. Again, it's because I don't want to. Well, I'm going to disappear for just a second here. But yes, SARF. Then SARF Lab is the working at the intersection of physics, biology, and computation to understand brain organogenesis. Right. So. SARF Lab seems like a good one to get in touch with too, and SARF seems to be joint appointments at Santa Barbara and Santa Cruz, and and it links that, and so like I mentioned and some of this is got a kind of renewed focus or energy because of the Biopunk Society, which is just a funny name. But it's very much it's a community biolab that started in San Francisco. I went to the opening, and yeah, and you can see it on the Slack under brain organoids. Yeah,
Yeah.
but the Biopunk Society. Well, anyway, they've got money from the city to have this space, but they're also going to be looking at other opportunities to kind of bring in some money or some like they seemed very interested in making links to labs, and so this kind of remote access, and I still anyway, I think it would be great for the city to see that they're making connections to academia as well. Right?
Yeah.
It's not clear to me what our kind of wet lab project would be.
Right.
Mainly because they just don't have the facilities.
Right.
I'm like, okay, you got pipettes,
Yeah.
you got lab desks, you got benches and pipettes. Like that.
Yeah.
Does not a lab make? They got some hoods.
Right.
But it seems like it's really early days, and what I really like about it is that it seemed like a lot of the people there. San Francisco has a huge biotech community, though, right? So it was a lot of people that came that were like, yeah, I work on, pick your thing, right? Protein synthesis, antigens, or the robotics that are used in, these kind of super high tech biotech facilities, right? Whether they're providing, lab services as a service, or whether they're, just doing it at kind of big pharma scale kind of things. A lot of people with a lot of good knowledge and skills that were really interested if there was like a good educational project or community or things to be involved with. So it like that's kind of exciting. Yeah, and so hopefully more. Well, it'll probably be. Not so much more before the holidays, but it'll probably be in January where we start to actually hear anything. And noted already that January would be the time Stanford does its like winter. I forget what they call it. It's like winter school for organoid protocols or something like that.
Oh, wow.
Like they have people come, and I'm still pushing. Well, regardless of whether they finally release their educational materials, like the great thing is the Brain Engineers group is totally set up to do that and to make everything available for others. So that'd be my update.
Oh, good. Yeah, I saw you had a flurry of activity in the Slack in the Brain Organoids channel today. So
yeah, just it's kind of replying to this person who's setting up Biopunk Society, or kind of leading it, and it's kind of grant and fundraising efforts. So
good.
That was it was nice.
Yeah, that's great. We've been trying to get that off the ground in some way for a while. So
yeah, and then again, like hoping that and then just the exciting thing, at least paper-wise, regarding that is the new work from Austria that is super cool. I mean, one, it's like, hey, we're modeling the dopamine system, but that's cool. But it's cool because they're doing 3 different tissue areas, and then they're linking them together, right?
Yeah.
So it's what we were talking about, which is like cerebral organoids aren't mini brains, right? They're an artificial tissue differentiation for a particular type. And so you currently have to do these separately. And if you want to kind of put together a whole circuit, right, that you're going to have to do these multiple preps, and then the harder task of like somehow getting them to grow together,
right?
And so that was an exciting paper to see. And it's from the original. I don't know how to pronounce it. It's like Noblich, but it's the PI that Madeline Lancaster was with when the first paper.
Yeah. All right.
Okay. That's a lot of exciting stuff. Okay. Before we get into the coding paper again, I'll mention one thing really quick that I'll go more in depth on Saturday. But anything from Amanda you wanted to say before we go into all this stuff?
All the organoid stuff sounds very interesting. I opened a bunch of the links that you sent, and I don't think I've really read any of them, but I want to. I don't think I have any substantial updates.
Okay.
I guess I can say now that I would be available to meet next week if we want to.
Okay. Yeah, I was going to ask about that. Let's. Is that good for other folks, or even if I may even just have a small meeting with? If you guys can meet, that's great too. I'm up for meeting. Actually, Wednesday should be fine for me. Yeah, Wednesday's good. Other days not so much.
All right.
Okay. Well, we'll aim for 8:30 start time like usual and see if that changes. So
Yeah.
we'll,
Do you want me to do
one
the?
more.
You want me to do
Sorry.
this thing I was going to present on before you do the coding paper, or do you want to do that first?
Yeah, we'll go through that. Why don't we do that? I'll very briefly mention this article just because I think it's we're talking about healthcare and medical stuff.
Yeah.
And much more in depth Saturday. It basically the short version of this article, the TLDR is, as we know, the FDA is supposed to regulate medical technology. We're kind of in a place where that's essentially impossible to beat the hot take, because AI, we can't tell them what to do, but we can educate them. So Google is not educating Congress on how to regulate Google, and I feel like that's that's what it is. The selling the AI Google selling is in a regulatory gray zone. The FDA authorizes AI-enabled medical devices, but the reviews were designed for less advanced technology. No one from the government is ensuring newer software-based tools do what they promise. I don't think this is a surprise to us, but it's just sort of like interesting to put out. There's a lot of like a consumer market. There's a lot being put into smartphones. So I think this, what we're saying about like we've talked a bit about like the Muse headband, the Mendy headband, these sort of consumer grade, easily accessible EEG devices that can measure different biometric kind of things. Obviously, you can get some EEG stuff. Maybe we'll see how what we can do with that. Hopefully in the future, and just like the advent of what you can do in the smartphones now that'll affect consumer stuff, but also it makes certain research avenues potentially more available to people like us. And then a lot about privacy. Marx is worried that HIPAA, decades-old law now designed to protect patient privacy, falls into a large language model, falls apart in the face of LLMs. Because it allows health systems and their vendors to use de-identified patient data. But I think we've doubled down on reporting, but not explained compliance. uh, what is the AI version of Hippo? Yeah, we'll put that away into the Society Ethics Tech channel. Hey, speaking of Nick Wick's stuff has been extended by a month, basically. So, you know, unfortunately, or fortunately, depending on how you want to look at it, with all this stuff and the momentum, I may submit something after all to them,
Okay.
especially on this topic, because it's like getting at the policy side of things and this stuff would be interesting. So more about that later. Bradley has generously volunteered to go into the why animals need a brain. It's an important topic.
So, last week I talked about this. I mentioned that why do we have brains if single cells can process information? And, why do you need like a C. elegans brain even if paramecium can do it in a single cell? And I didn't realize it at the time, but that was actually a chapter in a book by Peter Sterling, who does a lot of stuff on allostasis. And so let me see if I have it here. Principles of Neural Design. And yeah, there's not much to see. It's just kind of like one of these academic covers. But yeah. But it was chapter 2 basically talks about that very thing. And so that was exactly our conversation last week. So I'm going to go to the it's a kind of like a reprint in Animal Cognition of this chapter. So you don't need to have the book. This was published in 2015, this Principles of Neural Design. So this is, I'll kind of walk through this paper, which is chapter 2 in the book, and we'll kind of go from, we'll kind of think about why they say, and I don't know if it's the definitive answer, but why they say we need a brain. So the abstract in Principles of Neural Design, this is 2015 MIT Press, inspired by Charles Darwin. Stirling and Laughlin, Simon Laughlin is the co-author, undertook the unfashionable task of distilling principles from the facts in the technique-driven, data-saturated domain of neuroscience. So their starting point for deriving the organizing principles of brains are 2 brainless single-cell organisms. So they not only looked at paramecium, but also E. coli, which is a bacterial cell, and they live in colonies. And we know E. coli from making humans sick, but they also form colonies, and they do a lot of things that paramecium also do, like chemicals, sensing chemical gradients, and behaving collectively and things like that. And then, of course, they use the 302 neuron brain of C. elegans. So they use these and they put them into this framework of looking for principles. And so that's what the whole book is about. Chapter 2 is about sort of establishing, why we maybe have, non-neuronal cognition, why we have small connectomes, and, why do we have small connectomes if we have these single cell competencies. So the book is an exemplar on how to connect the dots between simpler and much more complex organisms in a particular area. So the question they ask is, why do we need a brain? So we obviously want a brain. Why do we need a brain? One's first thought, of course, is that we need it for the magical activities and feelings it confers: love, music, art, consciousness. But also these features arouse intense curiosity. They are merely Baroque decorations on the brain's fundamental purpose. It should not be mistaken for the purpose itself. And they talk about purpose. But, this is, so they kind of give some statistics here. So they start with the prokaryote, which is E. coli, lives of the brainless. And they talk about bacterium foraging. So E. coli being the size that it is, 1 micron by 3 microns, which is very, very small. Each cell kind of lives on its own, but it also lives in a colony, but they also have to find food. And so they thrive in a nutrient soup adrift in the intestinal digest of large animals. The microbe is equipped with taste receptors, a battery of proteins, each of which specifically binds an attractant, such as an amino acid or sugar or a repellent. So they do this sort of chemosensation where they pick up signals, they pick up things, in a certain direction, they pick up concentrations, and they can do all that with, taste receptors, which are basically these protein complexes that allow them to process this. And then these receptor proteins cluster on the surface membrane and form signaling complexes. So these signaling complexes then can change the sensitivity and response speed. And so this is basically where you have your sensory organ and your sort of everything forward from the intraneuron sort of in one complex. And then it can actually, the E. coli can actually integrate that information in some way and act on it. So it's acting upon information, these chemical gradients. Now, C. elegans also does this, but they do this with separating out the sensory organ from the circuit that guides movement, that guides sort of the sensing concentration and things like that. So this is all, they do the same sort of behavior, but they do this in a different way. And so, although each cluster comprises of thousands of molecules, to increase the chance of catching a taste, there are only 5 types of receptor molecule, each responding to a range of related compounds. E. coli uses glucose. When glucose is off the menu, it can use lactose. This requires lactose detectors to call for 2 proteins, a permease to emit lactose, and an enzyme, galactosidase, to split it. The genes coding the proteins are adjacent in E. coli's DNA. So this is where genes are coding for these, different enzymes to be produced to allow it to process these things. So, we have this, again, we have these very simple processes, and, we don't have a brain yet. We have these molecular complexes. So we have this, because we have a molecular complex, we also have these repressor systems. The repressor is the lactose detector, which, upon binding allolactose, an isomer that always accompanies lactose, changes shape and releases from the DNA. This allows RNA polymerase to move off and transcribe the operon. So if you've heard of the lac operon, this is what they're talking about here. So they use this genetic circuit, and they use this protein complex to sense things and then to, turn on and off this circuit. And so what is the effect of this is that the lactose receptor predicts for the organism what it will need to exploit, or what it will need to exploit this new resource. So it knows kind of after, with these two things working together, it can not only find resources and, have, deal with them appropriately, but also predict which ones will be coming next. So this is an interesting system. And it's well known because people have studied this for quite a while. And so a given level of lactose in the soup being the environment, calls for the proper amount of permease, which is matched by the proper amount of galactosidase. This design principle, matching capacities within a coupled system. Okay, so this is something we've seen this before, kind of with Mike Levin, where he talks about competencies and different competencies. This is matching capacities within a coupled system. Is the key to the organization of multicellular animals when it is called simorphosis. This is from Weibel in 2000. So there's a citation down at the bottom, which is simorphosis on form and function in shaping life. Okay, now I can read it. And this is a book. I've not read this book, but this is what they're getting at here. Okay, so let me go back up. It looks like I have some references from after the book was published too, I just noticed. But okay, so we're up here. And so we see here that simorphosis begins in the single cell. Okay. So this is where we have this process in a single cell. We don't need a brain. We just have these, like a gene circuit, and we have a protein complex, and it's able to do this sort of stuff. And it's just basically regulating. So, Peter Sterling is a big fan of, this idea of allostatic drive and allostasis. And so what we're seeing here is actually allostasis in the single cell, and, a response to that, the sort of the internal milieu of the cell and the outside environment. Okay, so that's kind of going through, talks about a molecular control system which combines information from two inputs to compute the correct conditions from processing lactose. So now this is where he gets into this, how this is sort of processed as information. So he's proposing that we have these basically logic gates which operate on the principle of the lac operon. So the lac operon is a system of regulating the system using gene expression. So it's a gene regulatory network, basically. And you have these different rules for triggering things. So how do you compute the correct conditions for processing lactose? There's if lactose and no glucose, then go. That's the first rule. So if you sense lactose and no glucose, then you turn the circuit on. And then the second rule is if lactose and glucose, then no go. You keep the circuit off. So you can see here that we have this configuration. We have different combinations of glucose and lactose. We have lactose only, no glucose, no lactose, glucose only, and glucose and lactose together. So you can see that it's on in the case of lactose, but in these other cases it's off. And that's basically how it works. So it's very much building on this idea of a logic gate and a logic circuit. So we can break this down, and people have done this in systems biology that work on these kind of problems where they build a gene expression network or gene regulatory network, and they build these logic gates that will allow for certain things to happen. And of course, we think of logic gates also in terms of neural networks, and so and in connectionism. So we have this part of it as well. We can do those kinds of things just in one cell. E. coli also has a working memory, and it basically needs to preserve the internal milieu and reproduce. To do that, has a working memory. E. coli's working memory is simple. It is imprinted on the receptor protein by means of a negative feedback loop. So this is a negative feedback loop with the receptor protein being involved. The activated taste receptor causes an enzyme to attach methyl groups to the receptor complex, decreasing its sensitivity. The number of methyl groups on a receptor indicates how strongly it has been activated. And because the feedback loop is sluggish, the record stretches back into the bacterium's frantic past. All right, so that's basically it. Basically, it establishes some sort of memory. So it's not, exactly what we would expect from, say, like humans or even, E. coli, but it's definitely a mechanism that can, accumulate information over time and retain it and recall information, So the mechanism, by using the past receptor sensitivity, determines the bacterium's response in the present. So this is their definition of memory, and basically does all those things that a larger connectome can do. Thus, a single cell can store information cheaply through chemistry by covalently modifying a signaling molecule. So, this is basically what we're doing with. So E. coli has this working memory, and it's doing this sort of, it's allowing for a biased random walk that allows maybe this output behavior that looks intelligent. So it can do a lot of things in a single cell. And then, you have protozoa, bigger and faster but still brainless. This is paramecium. And so this measures 350 by 50 microns. So it's a bigger organism. It's a 10,000-fold larger than E. coli. It's not as subject to viscous forces, so it doesn't have to deal with the sort of that regime. Paramecium propels itself with cilia that covers its surface. It can coordinate their beating to send synchronous waves from head to tail. In its cluttered environment, paramecium encounters immobile obstacles and avoids immovable obstacles and avoid the futility of continual ramming. So it has this response to sort of not, collide with obstacles. So it has sort of this obstacle avoidance mechanism. But it doesn't have a brain, of course. So what is it doing? So it's actually evolved a useful response, and this has been studied since this paper by Jennings in 1904. At the first bump, it throws its cilia into reverse and backs off by a few millimeters. Then it does a quick twiddle, switches to forward, and sets off in a new direction. So it can do this not only predictively, within a few millimeters, but efficiently as well. E. coli's chemical signaling systems could not trigger and coordinate this rapid response. In E. coli, diffusion suffices because the distance is short. So if it's a very small local environment that you have a small organism, you have a small local environment, you can use diffusion, which is like where things diffuse randomly, but because you're so small, it's something that can be used for, local sensing. And so because we're in a larger regime, we can't rely on that sort of thing. So we need to actually be able to trigger and coordinate a rapid response in other ways. And so diffusion time increases as the distance is squared. So if in bigger systems, you can't really rely on that. So in diffusion from head to tail in paramecium is something like 10,000-fold slower than in E. coli, so it's about 40 seconds. So that's quite a long time for things to diffuse through the cell of a paramecium. So this is where we have, in E. coli, we have these molecules inside the cell that are like diffusing and they're triggering processes. In protozoa, you need to have something where you have a formal pathway, maybe like, I wouldn't say like a digestive system, but some structure internally that you can have. So this is basically going through, basically this organization from head to tail, and it's not a nervous system, but it's not even like a coordinated set of nerves, but it is like the system that can coordinate different parts of the body to do things. So, if receptors at the head need to send a message to the tail cilia, it can, diffuse some molecules which diffuse down to the end, but that takes too long because you want to trigger the behavior quickly. And 40 seconds is a long time, especially if you're at that size scale. So, you want to spread your signal. You could spread it with an axon, but that's cheating at this point because paramecium don't have axons. So how do you do that? And so you have this electrical signaling system or this electrical signaling mechanism where you can change the membrane voltage initiated at the head, and it can reach the tail in milliseconds. And so electrical signaling, but to implement this electrical signaling in paramecium, evolution requires several new components. So first, the mechanoreceptor is needed to detect the bump. You get a mechanical sensation at the front. This involves a specialized caton channel inserted into the cell membrane. Stretch on the membrane deforms the channel. It opens sodium ions that rapidly depolarize the membrane. So this is where you get this quick process of depolarization, like in a neuron, except you're not in a neuron. And of course, we see depolarization in other cell types as well. We've talked about, different types of electrical activity in different cells. It's all basically based on the same principle of depolarization of the membrane of the cell, based on some set of ions that are coming in and out. So in this case, we depolarize the surface of paramecium. It can send a message from head to tail without having any sort of nervous system equipment. Okay, so this is kind of talking about thus multicellularity follows the engineering principle complicate. And this is from Glag, 1969, 2009. And this is a quote from this work. The many tasks performed by a single cell are now divided among many specialized components. Naturally, coordination is required at each level, which is cell, tissue, organ, system, and organism, and across levels. So coordination demands some mechanism with an overview that enables it to weight alternatives, set priorities, and then exert ultimate authority to execute. Fortunately, the multicellular design that demands such integration also provides a special class of cells to accomplish it. And so these cells, neurons, now do what paramecium could not, provide multiple fast lines for communication. So this is where we have these communication lines. We don't have axons yet, we don't have synapses yet, but we have this system of communication. So in short, for a multicellular organism, the brain becomes a necessary, possible, and profitable endeavor. So you have this, you have other types of communication you can use. You have these electrical pulses. But they only work in a single cell. So if you have a multicellular organism like C. elegans, you need to propagate that signal across cells. And that's not easy. You have, gap junctions, but those may not work for what you want to do. And then, you want to have some response mechanism. So you also need other things that coordinate those cells to do those things. So let's see. The electrical pulse generated by paramecium's ion channels provides another design principle. Combine analog and pulsatile processing where necessary. So pulsatile means you release something like you see in E. coli. Analog means you have this sort of depolarization like you see in paramecium. And paramecium's avoidance response, which depends on rapidly coordinating a large number of cilia spread down its body, is an example of the necessity of pulsatile processing. So you have this, you have to coordinate these cilia as well as send the signal. And so this is where you get this pulsatile processing. So that's, and that's where brains emerge, because paramecium's behavioral repertoire is limited. You can't do everything in this way, even though it's an improvement upon E. coli. And so now they get into C. elegans, connectome, and this is where they talk about sort of the multicellularity of C. elegans and some of the avoidance reactions that it has to different stimuli. Well, oftentimes it send its head in different directions, and if it finds a spot it doesn't like, it backs up and it kind of moves in a different direction, or does like these omega turns. It does all sorts of things where it can move adaptively through the environment. It can avoid certain chemical concentrations or certain barriers or whatever. So, C. elegans can do a fair number of behaviors. A lot of them are stereotypical in terms of movement, but they have now a nervous system that allows it to coordinate the cells in the body, coordinate the signals being sent throughout the body from like some sensory organs, coordinate the response to different types of sensory stimuli, and coordinate a behavioral response that plays upon that signal. So they talk here about a sequence of muscular contractions producing a moving wave, and you have other, so you have a formal connectome. It's sending signals to muscles. The muscles are moving. The muscles are moving in response to something that was sensed in the environment. So you have a sensory organ. So you have these sensors, these neurons, and then these effectors. And they can, they have this oscillation that that you can build an oscillator model where they kind of move back. They can oscillate back and forth. So sometimes they'll swim in media. Sometimes when they move, they oscillate back and forth. And that's how, they can maintain their movement. And, they don't need a lot of instruction other than having that system in place. So this is, your combining feedback from the environment with body mechanics. So if your movements are in a good, movements are enabling something good, then you continue to do it. If not, then you can stop doing it or you back off. And that all is, it's this sort of adaptive mechanism. So by using its biomechanics, the worm can dispense with a central pattern generator. So in insects, you see a central pattern generator where you have a single neuron that generates an oscillation, and it's usually in like a insect's legs, where they move their legs up and down, and there's like a small set of neurons that control that oscillation. So it's like a circular circuit that just kind of fires according to a pacemaker, and it's generating this oscillatory output. And but that's not really adaptive necessarily. You need something a little bit more for C. elegans for some of these other behaviors that it does. And so by using its biomechanics, which we have these sorts of mechanisms that build off of feedback and some of the muscle movements and some of the other aspects of the body. The worm can dispense with the central pattern generator, thus freeing up brain space. So when you don't have a central pattern generator, when you use an oscillatory circuit connected to muscle, connected to the environment, you don't need those kind of cells. You can have other types of cells that actually allow you to have more advanced processing. Here then is a useful design principle for motor systems. And here we're dealing with a sensory motor loop where we get sensation, we process that information, we send an output to muscles, the muscles move in the environment, and then there's a feedback to the organism to say, am I moving in the right direction or not? And that's a loop that's, closed loop system. And so this design principle is lighten the brain's load by using the body. So we know that from some of the stuff we've read about offloading, basically the idea of embodiment, which is that you use the body instead of the brain, or that the body is very important. So engineers call this embodied computation, also embodied intelligence or cognition. And this is the Pfeiffer and Bongard book that they cite for this. Make the paper available to people so that they can read it in more detail. But that's basically the idea of why we have a brain, because we do have to have these different competencies. And I don't know, maybe that's not the best answer, but it's an answer, and I think that it's pretty good in terms of thinking about some of the why do we need, why can't we just be one single cell? Why can't we just coordinate all our cells in a way that's promotes intelligence? And I think they give pretty good answers. Yeah, Jesse said PDF is in the Slack. Yeah, I put it in the Slack.
Yeah, Amanda also, I'll share my screen. Amanda generously donated the book to the Slack, to the Notion as well.
Oh, okay.
Morgan had just asked about it.
Yeah, I in events discussion, I put a link to a playlist of talks that happened at UCLA this summer. Yeah. For the was called "Pushing the Boundaries." Was the title, and then the subtitle was like, let me see. Oh shoot. Sorry, I forget what the subtitle is, but the main thing being that it was a bunch of talks that related to, like, a lot of this kind of material. One of whom I think it was Pamela somebody.
Lion.
Pamela Lion, yeah, who was just like started out her talk by saying like this particular Sterling and Laughlin book, is not appreciated enough, and but again, a whole bunch of talks regarding kind of like Levin's ideas about agential matter, just how much cognition is happening at from chemistry and fluids
Yeah.
to single-celled organisms, other species, like archaea, fungi,
Yeah.
like the different what am I?
Types of
what am I
single cell
blank-
organisms, I guess. Yeah.
Yeah, but phyla.
Phyla, I guess. Yeah.
Yeah. Sorry, I'm struggling for my vocabulary here.
Yeah.
But it's great. I mean, it's they start with Friston. The first talk is Friston talking free energy principle.
Yeah.
But then it's a bunch of other people that I learned a lot from, and and Richard Sully there. But I liked in particular, or I liked this chapter, going through it, and what they were saying. But I also like some of the speakers talking about intelligence from the kind of Tinbergen perspective, or like what constitutes how to classify animal behavior. And then I think one of the talks is trying to, do you see that in slime molds and fungus, etc.
Oh yeah.
And,
So it would be like Tinbergen's 4 questions, but applied to like.
Yeah. Thank you. Yeah. And so yeah, a lot of good ones. A lot of just cellular. Biochemical network stuff too, gene regulatory network stuff, etc. Yeah.
What was this event? Pushing the boundaries.
So if you go to the first one, so it's like the intro, and I didn't recognize the yeah. There you go. Welcome remarks. Yeah, that's funny. But there you go. Pushing the boundaries, neuroscience, cognition, and yeah.
Okay, I remember Levin talking about this a little bit.
Yeah.
I guess is that UCLA?
Yeah.
Yeah.
And I didn't recognize the a lot of them thanked. I think his name's David something. A lot of them thanked him for being invited. And again, like after I went through these, I was just like, yeah, this is an incredible mix of people. For what I think of as like Levin ideas, I see is actually a much broader community of people. So I think Amanda and I liked a couple of the, I'm gonna guess, Hungarians, just from the names and the number of accents on them.
Yeah.
But those are some interesting talks.
One of them talked about the concept of the cenome, like the sensory film. It was a bit quick, but I thought it was an interesting concept. That was,
David.
I don't know the name if I saw it.
Yeah, David Glansman is the person who put this together. Do you know that name, Bradley?
I'm not sure. I don't think so.
Yeah, I mean, there's plenty of people I don't know, but but I really liked this group, and they all seem to be deferring to like they said they were invited by him.
Okay, yeah. Okay, UCLA. Wow, that's certainly good. Yeah, where was this?
Yeah, the František Baluska, cellular basis of sentience and cognition. I think that was one Amanda and I saw.
Oh, was the cenome?
Oh, is that the scene? Yeah. There's another one. Again, like
cenome is sensory, or
yeah.
Oh, yeah.
Anyway, totally worth going through for sure.
Yeah, they had some things on RNA biology and combinatory logic, and
Yeah.
and slime mold stuff. So that's good.
Right. Slime mold stuff, and and that just also made me think of the kind of meetings that Adam Adamansky puts together.
Oh yeah.
Oh yeah, and one of the last talks, and this came up just because Amanda and I have been talking about EEG analysis, and especially like sleep and anesthesia. But Eric Winfrey, which is a name I hadn't heard in a long time.
Yeah.
And so that's
Eric Winfrey. Yeah, this is great. So that chapter was like exactly what I was interested in, like that question, and then so
perfect.
Did we have any other comments or questions that we wanted to cover on that article?
Thanks for going through it.
Yeah, no problem.
Yeah. I mean, it's still. I mean, I guess it was nice to see what they were saying about because I'm always thinking about like what Levin's saying that animals are using electricity for non-neuronal stuff, and so I liked the at least start of this in terms of when you just need to get a message around the cell fast, like the same reason we have telephones. It's electricity goes quicker.
Yeah, and it's not. And I think the point that they mentioned about diffusion was important. That things can diffuse, and of course we have like diffusion. We talked about the peptide connectome, where you have peptides that get released throughout, and then you have to sort of integrate that. So you could, I think there's a hypothesis that says that early life they were using peptides for communication, but that's not focused as much as you can get with neurotransmitters. So when peptides predominate, we use peptides. I mean, mammals use peptides today, for example, in neural signaling, but it's like when you use that as a predominant mode, it's not as efficient and it's slower because it has to diffuse outward and it's not as specific to the target. So, when you have systems like that, you have to evolve new solutions. You maybe scale up in size, or you become like a multicellular organism. You need these new innovations to really kind of find new ways to do this. So.
Yeah, for sure.
Yeah, I think this would be. I feel like the easiest thing to say is like, yes, thank you for going to that. There's a lot from the article and the book and now this conference that, like, I would like there to be kind of something. I don't know where this fits, like in orthogonal lab, but like I've been mentioning like sort of the diverse intelligences stuff, which is a little bit different, but like I would like there to be there should be. I mean, it fits with stuff. It fits with like even like developmental brain brick stuff, like going all the way back to that. Like, there's, I don't know what to say, but I'd like to center some more work, like something specific in this space, for next year. And I don't know how to do that, but open suggestions.
Yeah, I think something like a diverse intelligences might be a good theme, or like something like looking at all the different model organisms that we talk about and different types of systems, and then linking that with like intelligences or something like that.
Yeah.
I'll say it's almost like a survey of computational cognition, just like a project that I started up like years ago and never did anything with. But yes, that would be good. You were saying, Morgan?
Well, I was gonna say that like I think before we read the 11 paper like a couple weeks ago on the stability of memories, like Amanda had a like 4 11 papers put together. It was like maybe just an image of 4 11 papers put together.
Yeah, it was a screenshot from one of his lectures when he was like, there are interesting philosophical questions here, but that's not the point of this talk. But here are some places where I've written about it.
Here's one of the many other dimensions that I work on. Yeah. But those 4 papers, I think I said something like, these were the diverse intelligence papers or something like that. Like I'd love to get through those or discuss those together. That would be great.
Yeah, that would be good. I can try to find that screenshot.
Yeah, if you find it. Did you drop it in the Notion like maybe the Notion of that?
Oh, I found it. I shared it in the cybernetics reading group page, which is kind of sparse, so it's quick to find.
Sure.
Oh, okay. I can get that.
I got it.
Yeah. All right. Here we go. Well, there we go. Oh, you got it. Thank you, Amanda. Here we go. Interesting. Okay. So we'll just make a 11 fan club as part of our organization somewhere. Okay. We have about 20 minutes left. I think that might be enough time to finish the coding paper at last, if that's what you all feel like you want to do.
Yeah.
Sure. I meant to try to read some of the replies, but I did not.
I haven't really gotten. I looked at a few several weeks ago. I don't.
It was also nice to cover that chapter 2 of the Sterling Laughlin because it made me think of the Brette swimming neuron paper, like his eLife paper, or like computational modeling of the paramecium.
Yeah, I think the responses for this paper deserve their own session. I mean, we should just spend a whole session kind of going through them, but yeah.
Going through the responses, you mean?
Yeah,
or
the
like
responses. Yeah, because there are a lot of
them. Yeah, and like, I guess, I think we were kind of at the conclusion anyway. So I guess we'll just look at this section really quickly. I think we talked about blood pump before even, but it's talking about function, specific ways in which elements of the heart interact by identification of elements of the pump. In effect, the pump is a model of the heart. A metaphor is not simply words arbitrarily chosen to designate the object, but it is a model of the object, like Goff and Jackson, and as such, it deserves scrutiny. Another model of science is coding a good model of brain function. He talks about his 3 aspects: correspondence, representation, and causality. Technical results are based on the first aspects, but maybe not the other 2. It's the same amount of scrutiny. But why is evidence that such no representations exist? Why is the evidence why is the evidence that the brain can manipulate spikes in this way? Technically, it's found the activity of many neurons varies with stimulus parameter. but also sensory, behavioral, and cognitive context. Neurons are also active in the absence of particular stimulus. It talks about correspondence. Behavioral significance is only obtained by making an implicit linking proposition that relates encoders and decoders. Critically, the contextual dependence of neural codes can be solved by incorporating contextual variables in broader neural code, because this context is precisely what defines variables. Okay, properties of what? And I think we had quite a lot of discussion in the last few things about like properties of what, and is, you know, how much is this, how many layers do we need of encoding and decoding, and does it have to be separated out that way? Notion of information implied by the coding metaphor is inappropriate, is his argument. So, you know, a more appropriate notion is the formation of information as organization, namely relations between sensory signals and actions forming structured internal model. Disconnected from causal structure. Yeah, that's the reason for the paper. In conclusion, the causal structure of neural coding metaphors is incongruent with the structure of the brain. And what else if not coding? So I guess this is what else? Taking a shot at Cartesian philosophy again. Not context free, but symbols provided in neural codes are not context free. They're unstructured and no causal powers. They do not have, etc.
So this is interesting that we have this idea of a genetic code or genetic program, and people have similarly criticized that approach, because, you know, there's this idea that like there's a code in, you know, like there's obviously like the base pairs and you have codons and things like that, but there's this idea that like you have things that are coded in the genome. It's kind of a terminology people use. So like, you know, different traits are coded in the genome as like this explicit thing in the DNA. And then there's been a lot of criticism of that in terms of, is there a code or is there not a code? Is it just simply that you have, you know, like a string of base pairs that produce something like an RNA molecule and then produce a protein? And then that actually ends up giving you what you know, might think is a genetic code. So I like that, like, inclusion of that debate, although he could have unpacked that a bit more to kind of like talk about the parallels between neural coding and the arguments for and against a genetic code or a genetic program, because I think that would have been useful. I'm not, yeah, I mean, that would have been actually a useful paper in and of itself, but we're not in that space. I think he wanted to focus on the neural coding.
You said neural coding versus genetic.
Yeah, so up in this paragraph at the top of this page, the first paragraph, yeah, similar arguments been made against the idea of a genetic code or genetic program, and the alternative route is to adopt a systemic approach, which is where it's the genetic code or the genome is a system rather than like a code. And, you know, it's not just like, you know, a distinction without a difference. Actually, it does make a difference if you're thinking about what the genome can do, single genes versus groups of genes versus genes and environment. And, you know, that just influences that entire view. So it would be nice if like we had gotten more unpacking of that, but he cites Dennis Noble and then leaves it at that. And, you know, so I can't say that I have a really good appreciation of that debate, so I can't really add anything.
And he goes back into sensory motor systems, and all these people work on that. Go back to encoders and decoders. Methodology embracing behaviorism and dualism. Instead, it suggests developing models of the full sensory motor loop, models that behave yeah, not hierarchy, let's be this I heard. Yeah, okay. So I think that's sort of a nice summary. I think we kind of understand his viewpoint pretty clearly. Any discussion about the conclusion, or anybody? Is there any particular one that we wanted to maybe take a look at one of them, and then leave like 10, 15 left?
Yeah. So what's the Burkhardt 2015 C? This in particular has made a case for representations as a form of normativity realized by, what's that? 20.
I think it's down here. Here we go. Okay. Sure.
Okay.
Okay. Oh.
Okay, Journal of Theoretical Philosophical Psychology. Interesting.
So, yeah, this I think this is a good sort of concluding part here where he talks about, like, I was kind of thinking, well, what is his actual thing that he's advocating? And I think, he talks about internal models as being a good, like, alternative to neural coding. Then he talks, then that, of course, that paper, they advocate, I guess, for what's the alternative to computation. If you, so your internal model, what is that? It's this black box again. So the internal model then is maybe connectionism. So it's like some sort of network, or it's a dynamical system, which is some sort of like mathematical representation. Or, I guess we have other things in here that might be also, I think he talks a little bit about the free energy principle and kind of a smattering of things that exist. But I don't know, the models that behave, which is actually interesting. I've never heard of that phrase. That's Gomez-Marin, 2017.
Yeah.
Could you go down to that reference, Gomez-Marin?
Yeah. FG, another one. Fodor, Jerry.
Yeah.
What was
Gomez, yes.
Gomez.
G-O-M-E-Z.
Oh, excuse me. G for Gomez.
There we go. Gomez-Marin, 2017. Causal circuits explanations of behavior are necessity and sufficiency, necessary and sufficient. If you go to the citation.
So a chapter?
So I think this, it's a book chapter. And this is basically, I guess it's about causality. So in the current advent of technical innovation, allowing for precise neural manipulations and data collection, it is hardly questioned that the explanation of behavioral processes is to be chiefly found in neural circuits. And so he talks about I argue that to upgrade intervention to explanation is prone to logical fallacies, interpretational leaps, and carries a weak explanatory force. And so this is where, to claim that a behavior is explained by a necessary and sufficient neural circuit, which means you can like take parts out of the neural circuit and see if it still works or if it doesn't work, is at best misleading. And that my critique rather than criticism is deemed mainly negative. He wants to adopt circular causality, searching for principles of behavior, and embracing process philosophy rather than substance mechanistic ontology.
Process philosophy appears.
Yeah. So that's this paper.
Good. Sorry.
So yeah, it's basically, like we're not thinking about the link between brain and behavior properly. We're kind of manipulating circuits in terms of like addition and subtraction, so we can, take out a neuron or take out some functionality. We know like the experiments with HM where there was some sort of retrograde amnesia. You can actually do these experiments where someone's had part of their brain damaged or removed or something, and it can be, you can see an effect on behavior, but that doesn't always hold true. In fact, most of the cases of behavior may or may not be amenable to that type of analysis. The standard in neuroscience, of course, is the sort of, is it necessary to produce a behavior? Is something sufficient to produce a behavior? And what he's arguing is that's not enough really to say if some circuit is really producing or controlling a behavior. Okay. So yeah, we can go back to the paper if you want.
What? Just to ask the question, what is process philosophy?
The basic idea is that like the most. Have you heard the phrase "carving nature at its joints"?
Yes.
To refer to
I
like.
haven't probably mentioned that once or twice, but I'm still getting used to what it means.
Basically, picking out natural kinds, like picking out the categories that refer to like real things, having our delineations refer to like real separations between things in the world. So in contrast with process philosophy, you'd have like a substance-based ontology where like the things are the natural kinds in the world are what we would normally refer to as like things or objects or it's stuff. But in process philosophy, the most foundational like natural kinds are relationships, and they're like dynamic. There's a temporal aspect. So yeah, I think the easiest way to explain it is to contrast it with a substance ontology. Whitehead was the main philosopher, to my knowledge.
Yes. I would ask you more about that, but plenty of time and like your general opinion of it. But basically, I found a book that was very interesting by, I think his name was Cobb, and somebody else talking about process philosophy and basing all this stuff off of Whitehead. And I'm like, oh, I have to look more into that to see what I really feel about some of those things. But anyway, we have a few minutes left, so we can continue talking about this paper, I suppose. So this looks interesting. Is possible. It's not a question. Paul's saying it's happening. It's already here. Which is ironic, evolutionary, yes. I want him to go on that. I think we're looking at references and citations. Here we go. Any other citations we want to look at from the conclusion? We just hit Gomez Marin. I think that was the last one he mentioned before his conclusion here.
So, I mean, I guess my other question was, what does he actually advocate at the end of the day? And so I guess what he's saying is that I think there are these useful alternatives, but I don't know, and nobody knows yet. And so it's kind of a, coding is incomplete, but what takes its place or what's the alternative? And so that's, I mean, I guess so. There's this internal model. We have dynamical systems. We have maybe things like, you know, a free energy principle, or things like connectionism, or you know, I mean, even process. You know, it really depends on, you know, what the current flavor of the month is. So, and you know, that's the thing. Like, you know, we create a lot of a lot of approaches in science because we think that, you know, it's a good heuristic for trying to find out certain things about a system. So, you know, you could see where people would think, well, you know, coding isn't a bad idea. It's probably, compared to the alternatives, maybe the best idea we have, right? I mean, you have to take the good with the bad. It's maybe not going to serve very well as a metaphor, but it does do some explaining of what the brain is doing. It does give us a good framework. On the other hand, you know, it could be that those cons are just too much, and that, you know, we need to replace the idea. So I'm not I'm not sure. Like, I know that he's advocating for something other than neural coding, but you know, I don't know exactly what that would be or what that would look like. And it looks like the responses have considered that. So we should do that in another session, kind of like go through the responses and kind of hit a couple of the key ones and see what they think.
Yeah, I mean, I'd be up for that. I think maybe I'm not even going to say, oh, let's split up the number and look at 4 to 5 and see if they're worth talking about. I think we can just try to, if you find one, maybe mention it in the Slack, which would be really cool to say, hey, if this one, flag one as like, okay, let's definitely talk about this if you come across it. But yeah, I think that's fun. That'd be a nice, you know, get some, put on some holiday music and wrap up the year
with commentary from behavior and brain science.
What better way to end 2023 than understanding and discussing responses to a critique paper about the neural coding metaphor? That's awesome. Okay. Yeah. I mean, I'm for that. I'm sure I have some other things to say. Any other thoughts or comments before we wrap up for today? I'm sure the Paul Kai said will come up on this.
Yeah. Oh, I guess one last thing I'll say about the first part of the meeting is that, you know, we talked about the principles of neural systems with Peter Sterling's view, and then we kind of hit the last part of this paper. So is Peter Sterling kind of arguing for coding, or is he arguing for an alternative approach to understand the nervous system, or what? Because it isn't like the traditional view of neural coding. On the other hand, he starts getting into things like logic gates, and talking about how sensory, you know, protein complexes at sensory organs can transduce information. And so there are all these things that are maybe kind of like codes, but, you know, he doesn't say they're codes, but maybe he's arguing for something else. I don't really know.
Maybe it's a form of process philosophy with its relation to whatnot. I'll leave it at that. Thank you. I feel like everybody has masked how immensely fatigued and tired I am, and kept it. I like absorbed like 15% of what everybody said, and I appreciate the kind of work we did for that today. It was a lot of fun. I'm glad we can make it. Hopefully, we can try to make it all next Wednesday. Go from there. I'll have a few more things on Saturday to talk about. There's no Thursday meetings for anything under now, but that will pick up in the next year. So hopefully see everybody on Saturday and have a great night. And thanks for tuning in to 66th meeting. Awesome. All right.
Great.
See you next time.
See you.
Take care.
Take care.
Bye-bye.